Hi everyone, welcome to chapter 3. This is the chapter where we will be getting into cells, cell structure, and cell function. And I like to start this lecture by telling a little known story about this gentleman right here, Robert Hooke. Now your textbook mentions Robert Hooke, but it doesn't tell you the story that I am about to tell you. So Robert Hooke was a scientist in the late 1600s, and this picture that you see on the left side of the slide is actually a modern recreation of Robert Hooke. And so this is probably not what he actually looked like. There are actually no known existing paintings or portraits of Robert Hooke. And one of the reasons why is because of his bitter rivalry with another scientist that you probably have heard of, whose name is Isaac Newton. So these two men, Newton and Hooke, they were both competing to be the greatest scientific mind of their era, and the competition got very dramatic. They had these vicious fights via letter that they sent back and forth to each other. At the time, Robert Hooke was the president of the Royal Society of London, which was a scientific organization that Newton submitted his research to, and the two men often clashed in a very heated way about ideas and discoveries that both of them claimed were their own. And their rivalry it went on for over 30 years, and it only ended when Hooke died in 1703. And Newton then succeeded him as the president of the Royal Society of London. And at that point, he had Hooke's portrait removed from the halls of the Royal Society, and not just removed, but historians believe he also had it destroyed. That's how much he hated him. And then he went on to destroy Hooke's reputation as much as he could. And he did a great job of it, too, because most people have heard of Newton, but they've never heard of Robert Hooke. But even though his name recognition has pretty much been stomped out thanks to Isaac Newton, Robert Hooke was undeniably one of the most talented and impactful scientists of that time. Not only was he the first person to propose that light travels as a wave, and he also discovered the physical laws that made it possible to create the first accurate clocks and watches. He also constructed some of the most sophisticated microscopes of his time, and he used them to observe all kinds of tiny organisms. And then he compiled his observations in drawings and descriptions that he put together in a book called Micrographia, or some physiological descriptions of minute bodies made by magnifying glasses. And that book is what you see in the image on the right. And this was the first book to ever publish illustrations of organisms that were seen through a microscope. And here are some of Hooke's drawings that were included in that book. So in the upper left-hand side, there is a flea, and then below that, there's some mold. In the middle, you're seeing the underside of the leaf of a stinging nettle plant. And on the right-hand side, we are staring down the head of a drone fly. So as you can see, Hooke documented quite a wide variety of living things, as seen under the microscope. But the discovery he made that really embedded his legacy came when he was looking at a sample of bark from a cork tree under the microscope. This is the image that he drew of what he saw, and in his writings, he described the appearance by saying that it was perforated and porous, much like a honeycomb, and he commented on how there were these microscopical pores that no one had ever been able to see before. And what Robert Hooke was seeing for the first time ever were actually the cells of the cork tree. Each individual pore, each slot in the so-called honeycomb was a cell. So he was the first person to see them, and at the time, he didn't really understand the significance of what he had found. But in the 300 plus years since the discovery, biologists have come to learn a lot about cells and their importance in living things. So we now know that cells are the basic unit of organization in living organisms. And by this we mean that cells are the smallest unit that exhibits all 7 of the characteristics of living things that we talked about back in chapter 1. So you may remember seeing this slide where we talked about the organization of living things. And while cells are not the smallest level of organization studied in biology, because both molecules and organelles are smaller than cells, cells are the smallest unit that actually meets the standard of being alive. Molecules are not alive, organelles are not alive. So in our last chapter, we had talked about the molecular level of organization in living things. And now in this chapter, we will be moving up the organizational scale to talk about cells and organelles. Organelles are the tiny organs that you find inside of cells, so discussion of the two goes hand in hand. So what else have we learned about cells since Robert Hooke's time? Well, we know that all living things are composed of at least one cell. Everything that is alive that we know of is made of cells. Sometimes this really is just exactly one cell. So bacteria, for example, are made of just a single cell. But there are larger organisms like plants and animals that are multicellular, meaning they're made of many cells put together. We also know that all cells arise from pre-existing cells. And by this we mean that every new cell is created through a process by which an existing cell undergoes division and makes a copy of itself. And later in the semester, we are going to learn more about the details of that process of how cells give rise to other cells. So that lays some groundwork of what cells are. Now let's take a look at the scope of our exploration of cells. The questions that we are going to answer in this lecture are, firstly, what is the structure and function of cell membranes? How do materials move in and out of cells via their membrane? What are the general characteristics of the two major classifications of cells, which are prokaryotic cells and eukaryotic cells? What structures are found in prokaryotic cells and what are their functions? And then finally, what structures are found in eukaryotic cells and what are their functions? So let's get started by diving into that very first question. What is the structure and function of cell membranes? So a cell membrane is a very important structure that every cell in existence has. It's a layer of material that encases the cell. The membrane is what separates the cell from its surroundings. It's also called a plasma membrane because it has this semi-fluid, flexible consistency that is kind of like plasma. So here in these images on the left, you can see an illustration of a cell that has a slice taken out of it, which enables you to visualize what the cell membrane is made of internally. It's composed of a bilayer, or in other words, two layers of phospholipids that are facing each other. And a more close-up image of that bilayer is what you see on the right-hand side. So as a reminder, we talked about phospholipids in last week's lecture, and we talked about how they have an unusual chemistry where the phosphate head is hydrophilic and wants to mingle with water, while the tails are hydrophobic and do not mingle with water. And then based on this information, I posed a checkpoint question to you, asking you to imagine what might happen when we take these phospholipid molecules and place them in water. How would they react? So now is the time where we get to reveal the answer to that question. So we know that one end of the molecule wants to be in the water and the other end does not. So when you get enough of these together, these phospholipids will spontaneously organize themselves into an arrangement that meets both of those requirements. They will line themselves up in two layers with the phosphate heads facing outward and the fatty acid tails facing inward. And this happens on its own. Just like oil automatically, spontaneously separates from water, phospholipids automatically do this. And by doing this, the reason why they do this is because the phospholipids will use those phosphate heads to serve as a barrier that allow them to exclude water from the middle part of the membrane, which is where the fatty acid tails are hanging out. Because the fatty acid tails are hydrophobic and they want to be protected from exposure to water. And meanwhile, the hydrophilic phosphate heads are exactly where they want to be. They're exposed to water on the outside of the cell and also on the inside of the cell. So this phospholipid bilayer is what the cell membrane is made of. Two layers of phospholipids facing each other in a way that satisfies their chemical properties. And this is why those properties of water that we learned back in chapter 2, the fact that polar substances want to be exposed to water and non-polar things do not. This is why those were so important. They cause cell membranes to form spontaneously when you get a collection of phospholipids together in water. Now, while the phospholipid bilayer is the main material that cell membranes are made of, there are some other components that are attached to or embedded within that phospholipid bilayer. So here we see a more extensive diagram of a phospholipid bilayer and some different elements that are embedded or attached to it. The major thing that's embedded in the cell membrane is proteins. In this illustration, the proteins are shown in that light blue color. And you may remember from our previous lecture that proteins are extremely important to cells because they are involved in virtually every function that the cell carries out. So these proteins embedded in the membrane, they may serve as transport channels through which molecules move in and out of the cell. They may be enzymes that mediate chemical reactions that occur either on the inner surface of the cell or the outer surface of the membrane. They can also serve as anchors for other molecules, like carbohydrates, on the exterior surface of the membrane. So these carbohydrates that are sticking off of the exterior of the cell, they can behave like the cell's ID badge and serve as identity markers for recognition by other cells. The carbohydrates that you see attached to proteins here, they're called glycoproteins, and I've just highlighted one of them in a yellow box. But carbohydrates can also be attached directly to a phospholipid, in which case it's called a glycolipid instead of a glycoprotein. You may also notice that the proteins in the membrane are different shapes and sizes. Some of the proteins are fully embedded and integrated into the membrane, and these are called integral proteins. So you can see an example of an integral protein in the box on the right. So integral, you think integrated. Other proteins are attached to one side of the membrane, maybe just the exterior surface or maybe just the interior surface, and they don't pass all the way through the membrane. And these are called peripheral proteins. So that's the one that you see on the left. Think of your peripheral vision. That's the vision that you have at the edges of your view. The word has the same meaning here. Peripheral proteins are proteins that are located at the edges of the membrane. And then the cell membrane also contains molecules of cholesterol, which are located in the hydrophobic middle region. And the cholesterol molecule is now located in one of those yellow boxes here. And the molecules of cholesterol play an important role in keeping the membrane fluid and plasma-like, even while the temperature in the environment changes. However, one thing to note is that cholesterol is only found in the cell membranes of animals. It's not found in plants, and fungi have a relative of cholesterol called ergosterol in their cell membranes that serves a similar function. So now that we've gotten a sense of what the cell membrane is made of, let's move on to discussing how materials cross that membrane as they move in and out of the cell. Because the cell doesn't exist in a vacuum. In order to survive, it has to exchange materials with its environment. And the cell membrane has this very important role of regulating the movement of substances in and out of the cell. So depending upon the cell's needs at any given time, the membrane will draw certain substances inside and prevent other ones from entering or expel certain substances from the inside to the outside. And all of this entry and exit of materials takes place through one of two different types of movement. There's passive transport of materials and active transport of materials. And both of these types of transport are named pretty logically. So passive transport, for one, is defined as the movement of substances across the membrane in a way that does not require the cell to expend energy. It's passive. It happens naturally. No effort is required. The way that this occurs is through a phenomenon called diffusion. So diffusion is the natural tendency for substances to move from areas of high concentration to areas of low concentration. In other words, substances have a natural tendency to spread out throughout the space that is available to them. And I'm sure we have all observed this somewhere in our everyday lives. In this video clip, for example, what you will see is drops of green food coloring being added to a beaker of water. And you can see very clearly how the food coloring is initially concentrated, but then it spreads out and it's on its way to being uniformly distributed throughout the beaker of water. That is diffusion. Another example you may have experienced is if you're in a room and someone sprays air freshener on the other side of the room, then after a period of time, you can smell the air freshener because the molecules are spreading themselves out throughout the room, and eventually they spread out so much that they reach you all the way across the room. When it comes to diffusion in and out of cells, there are 3 different variations of diffusion. There's simple diffusion, facilitated diffusion, and osmosis. So let's talk about these 3 one by one. In simple diffusion, molecules will move from high to low concentration directly through the phospholipid bilayer of the cell membrane. It's kind of like they're walking through walls. They just weave their way right through the phospholipids to the other side of the membrane. And not very many molecules can move this way. Only small, nonpolar molecules are able to be transported via simple diffusion. Some examples of small nonpolar molecules that walk through the walls of the membrane are oxygen and carbon dioxide, which are the molecules we inhale and exhale when we breathe. Several important vitamins like vitamin A, vitamin D, vitamin E, and vitamin K, and also steroid hormones like testosterone and estrogen can move this way as well. So this being the case, and given what you now know about the chemistry of the phospholipid bilayer, in our first checkpoint, I'd like you to tell me, why do you think only nonpolar molecules rather than the polar ones can be transported across the membrane by simple diffusion? Tell me your understanding of why this would be the case based on what you know about how the chemistry of the membrane works. Okay, so if simple diffusion only works for small nonpolar molecules, what options do polar molecules have for crossing the membrane? One option is called facilitated diffusion, which is the second type of passive transport on our list. Facilitated diffusion is where molecules move from high to low concentration, not through the membrane itself, but through a protein embedded in the cell membrane. So these integral proteins that we saw a few slides ago, they can act as channels for materials to pass in and out of the cell. Some examples of molecules that are transported by facilitated diffusion are monosaccharides, amino acids, and ions of minerals like calcium, magnesium, sodium, and others. The third type of passive transport is called osmosis. And osmosis is the passive transport specifically of water molecules. It's a variation of simple diffusion pertaining to water molecules moving through the membrane. Now, if you've been following the past few slides, you might say, wait, simple diffusion is for small nonpolar molecules. And we learned in chapter 2 that water molecules are polar. So what's going on here? Well, it turns out that water molecules are so small that they can actually weave their way through the membrane without issue, despite the fact that they are polar. So water molecules are an exception to rule. They are polar, but they can also walk through walls. Another way that osmosis breaks the normal rules is that in osmosis, water molecules move toward the area where there are more dissolved particles. So we just learned that in diffusion, particles try to spread themselves out through the space that's available to them. And we saw this with the food coloring dissolved in water. But here's the thing. While those food coloring molecules are spreading out towards areas where there are fewer food coloring molecules, the water molecules surrounding them are simultaneously being drawn toward the area where there's more food coloring. So the mixing is happening in two ways. Water is being pulled towards the areas where there are more dissolved particles, and that facilitates the diffusion. So this animation shows you what that looks like in action. It shows a beaker with a membrane down the middle, and this is meant to represent the cell membrane. On the left side, there are water molecules. The little red and white particles are the water molecules. You can see them on the left side of the beaker swirling around. The left side is pure water. There's nothing dissolved in it. And on the right side is water with some dissolved particles. The dissolved particles are shown in teal. So the water is going to be drawn toward the area where there's a higher concentration of dissolved particles, which in this case is the right-hand side of the beaker. And this is especially true if those other dissolved particles are too large to cross through the membrane in the opposite direction. So those teal dissolved particles may want to spread themselves out to the left, but they can't cross the membrane, so the water comes to them instead. So that's osmosis, and osmosis can have important consequences for cells depending upon what their environment looks like in terms of dissolved particles. When the concentration is equal on either side of the cell membrane, meaning that there is the same concentration of particles inside of the cell compared to the outside of the cell, we say that this cell is in an isotonic solution. So under these circumstances, water is not drawn in one direction or the other. The cell's water content is in balance because the concentration of dissolved particles is also in balance. However, if the environment outside of the cell has a lower concentration of dissolved particles compared to the inside of the cell, so in other words, the outside is more dilute, then water will be drawn toward the area with more particles, which is the inside of the cell. And we call this a hypotonic solution because the prefix hypo means lower, and the area outside of the cell has a lower concentration of particles. So think of the word hypothermia. Hypothermia describes a situation where your body temperature is too low. So hypotonic means that the concentration is too low outside of the cell. So under these circumstances, water gets drawn to the area with the higher concentration, which is inside of the cell. And as a result, the cell will swell and possibly even burst if the osmotic movement is significant enough. The third and final situation is the opposite of hypotonic. A hypertonic solution means that the concentration of particles outside of the cell is higher than inside of the cell. The prefix hyper means higher. So think of hyperactive. Hyperactive means too much activity. So under these circumstances, the water will be drawn out of the cell to the area where there is a higher concentration of stuff. And as a result, the cell will shrink and shrivel and dehydrate, and this can also lead to cell death if the effect is significant enough. And I'd like to give you a concrete example of how the phenomenon of osmosis can have serious consequences for cells. So the headline here, woman dies after being in water drinking contest. Although this article is a little dated, this took place in 2007, this is the very real account of a 28-year-old woman who had entered a water drinking contest hosted by a radio show to win a gaming console, and she died as a result of drinking too much water. This phenomenon is called water intoxication, or the medical term is hyponatremia. And this is not the only case of this happening. People actually die every year from accidentally drinking too much water. And before you stop drinking water, let me reassure you that you have to drink a lot of water to be at risk for this. You have to drink several gallons over the course of just a couple hours, which most people would normally not do. But the point of me sharing this story is so that we can put it in the context of osmosis and see how the harm can occur. So when you drink too much pure water in a short period of time, what happens is you actually dilute your blood. And At the point where the bloodstream feeds to your brain, the brain cells then become exposed to a hypotonic solution that is more dilute outside compared to the inside of the brain cells. And as a result, water starts to move from your diluted bloodstream into your brain cells, which causes them to swell. And because of this, the first signs of water intoxication are usually neurological signs, like a headache, disorientation, muscle weakness, and eventually progression of the swelling can lead to coma and death. So this is just one real-world example of osmosis taking effect. Let's take a look at another real-world example in this checkpoint. When salt is sprinkled on the body of a snail or a slug, the slug cells rapidly eject water, which is what you can see going on in this image, where the foam is being produced. And often this kills a slug via dehydration. So explain why this happens. And then one more checkpoint. When a person who is dehydrated needs fluids to be given in an IV, the medical professionals don't just give them pure water. Instead, they give a solution that is 0.9% sodium chloride or salt. Why do you think it's important to have the salt in the liquid that's provided in the IV? And then lastly, in this checkpoint, we have an animation showing several different types of transport going on here. Based on what you see, I would like you to tell me first what mechanism the blue spheres on the right are using to cross the membrane, and then what mechanism the orange spheres on the far left are using to cross the membrane. And be as specific as possible in your answer. Instead of just saying passive transport, tell me what type of passive transport. And while we are on this animation, I want to use it to transition to our discussion of the second major way that molecules are transported across the cell membrane. We just finished talking about passive transport, and the second type of movement of materials is called active transport. That's what this animation is showing happening to the pink spheres. And the way that they're showing that it's active transport is that there's this little burst of energy shown in yellow moving up to the protein and being used to power the transportation of the pink spheres across the membrane. Because unlike passive transport, which uses no energy, the distinguishing feature of active transport is that the cell has to expend energy to carry this out. Active transport is necessary in a few circumstances. One of them is when molecules need to be moved in the opposite direction of what diffusion would dictate. So from low concentration to high concentration, that requires energy because it's the opposite of the natural tendency to spread out. Active transport is also needed to move very large molecules, as well as large quantities of molecules at the same time. Because in these cases, little protein channels won't do the trick. Instead, a larger scale operation is required to bring very big stuff into the cell. And the large scale operation is called endocytosis. Endocytosis is a type of active transport that is used to move very large particles or large quantities of particles into the cell. And the way that it works is that the cell membrane will actually change its structure to absorb these large objects. So the membrane will form a pocket, which is what you see happening in this animation, and then the large particle will enter the pocket, and then the pocket will fully close and pinch off. And the result is the creation of this little package of material surrounded by cell membrane called a vesicle. And these vesicles can then travel around inside of the cell to reach the destination where the cell needs them to go. Here is an actual example of exocytosis taking place under the microscope. So here we see an amoeba, which is a type of microscopic organism, extending its cell membrane to create a pocket and surround what is actually another smaller microscopic organism called a paramecium, and the amoeba is going to bring that paramecium inside and eat it. Cells can also use this very same process to expel large particles, or also large amounts of particles from the inside to the outside. And when they do this, the process is not called endocytosis, it's called exocytosis. So it's more or less the reverse of endocytosis. The cell will place the materials to be ejected inside of a vesicle. The vesicle will move to the edge of the cell and fuse with the cell membrane, and then the contents will be dumped outside of the cell, and now the vesicle doesn't exist anymore. The membrane of the vesicle has become part of the cell membrane. And again, I have an actual video of this happening for you to see. This is under the microscope, and this one is not very high quality, but what we have here is a paramecium, which is the same type of single-celled microscopic organism that we saw the amoeba eating in the last video. And on the very bottom of the paramecium, what you can see if you look closely is a vesicle emptying its contents outside of the paramecium. So that's how the paramecium is expelling waste. So at this point, let's take a step back and revisit the bigger outline of this lecture. We have looked at the structure and function of cell membranes and how materials move in and out of the cell membrane. And what's up next is we are going to dive into looking at the two major classifications of cells, which are the prokaryotic and eukaryotic cells. And then we're going to talk about the structures and features of each of these two types of cells. So, biologists divide all cells up into one of two categories. There's prokaryotic and eukaryotic. And here are the images that you'll find in your textbook to depict these cells. But seeing them side by side like this is a little misleading, because if we were to actually accurately put a prokaryotic cell and eukaryotic cell next to each other in a manner that was to scale, then this is what it would look more like. There is a significant size difference between prokaryotic and eukaryotic cells. There's also many other features that distinguish them from each other. One of them is that while all cells are small, prokaryotic cells tend to be much smaller than eukaryotic cells. They range from 0.1 to 5 micrometers in diameter. And to give you perspective, a micrometer is 1,000,000 times smaller than a meter. Eukaryotic cells are larger. They range from 10 to 100 micrometers in diameter. Perhaps the most defining feature of prokaryotic cells is that they lack a nucleus, which is the structure that houses the cell's DNA. Prokaryotic cells have DNA, but they don't store it in the nucleus, whereas eukaryotic cells do store their DNA in the nucleus. And along with lacking a nucleus, prokaryotic cells also lack organelles, which are internal structures bound by membranes that perform specific functions for the cell, whereas most eukaryotic cells contain a wide variety of organelles. Lastly, the types of organisms that are composed of prokaryotic cells are single-celled microscopic organisms, which includes the categories of bacteria and archaea. An archaea is a type of microscopic organism that we don't really hear much about, but they are sort of the lesser known, but also prokaryotic cousins of bacteria. On the other hand, the types of organisms that are composed of eukaryotic cells range pretty widely from single-celled microscopic organisms all the way up to large multicellular organisms. So for example, the amoeba and the paramecium that we just saw in the microscope videos a few slides ago, those are both single eukaryotic cells. And these little single cell eukaryotes are called protists, but eukaryotic cells also compose plants, animals, and fungi as well, which are, of course, larger and multicellular. So we want to tackle each of these categories of cells one by one, and we're going to start with the simpler one, which is the prokaryotic cells. And we're going to discuss their important structural features. So we'll turn our attention back to this illustration of a prokaryotic cell that comes from your textbook, and we'll walk through the items that are labeled on this image. But we're actually going to start with something that's not labeled on this diagram, and that is the cytoplasm. So all cells are filled with a gel-like substance called cytoplasm that is 70 to 80% water. Everything else that is located inside of the cell is suspended in the cytoplasm. One of these things is the prokaryotic cell's nucleoid region, which is the area where the cell's genetic material is located. In other words, it's DNA. The nucleoid region is not bound by any membrane or any barrier. It's just the part of the cytoplasm where the DNA hangs out. Moving to the right, the flagellum is a long strand of protein that's used for cell movement. It rotates, and as it rotates, it propels the cell through its environment. Not all prokaryotic cells have a flagellum, though. Only some of them do. Next, we have the pili, and these are somewhat similar to the flagellum in that they are projections of proteins that extend off the surface of the cell, but they're used for a different purpose. These pili are little tubes that the prokaryotic cell can use to transfer genes to other cells. And this is kind of a strange phenomenon for us to wrap our heads around because we as humans can't just walk up to another person and say, hey, would you like the gene for brown hair? And they say, sure. And suddenly they have brown hair. But many types of bacteria can do this using their pili tubes. And it's actually a problem from a medical perspective because bacteria have been documented sharing harmful traits with each other by transferring genes through their pili. Like, for example, genes that allow bacteria to create certain toxins or genes that allow them to be resistant to antibiotics. Next, we have the the cell wall. The cell wall is a layer of material outside of the cell membrane that provides protection for the cell, and it also helps maintain the cell's shape. In bacteria, the cell wall is made of a material called peptidoglycan. And then now we'll get to the capsule. In some bacteria, there's an additional layer of protection outside of the cell wall called a capsule. The capsule is made of carbohydrates, and its role is to provide that additional layer of protection, and also it allows cells to attach to surfaces in their environment. In many species, the capsule is made of a sticky material, and it facilitates the cells adhering to things. So if you have ever run your tongue over your teeth and you feel that there's like a sticky layer of plaque on the surface of your teeth, that is facilitated by the bacteria using their capsule to attach to the surface of your teeth. And lastly, we have the ribosomes. The ribosomes are the tiny little dots that are speckled in the cytoplasm of the cell. And although they are tiny, they are very important because the role of the ribosomes is to produce all of the proteins for the cells. And as we know, proteins are basically the workhorses that make all of the cell's functions possible. So that completes our look at the structure of prokaryotic cells. Let's wrap up our discussion of prokaryotic cells with a checkpoint. The goal of this checkpoint is to match the prokaryotic cell structure on the left to one of the descriptions on the right. So now we'll be moving on to talk about the structure seen in eukaryotic cells and their functions. And remember that in eukaryotic cells, there is somewhat of a wider variety than prokaryotic cells. Eukaryotic cells are the types of cells that you find in plants, animals, fungi, and protists. So there will be some variation where certain structures that we learn about are found in plants, for example, but not animals, or in fungi, but not protists. But one thing that is universally seen across all types of eukaryotic cells is that, like their prokaryotic counterparts, eukaryotic cells contain cytoplasm, which is the gel-like substance that's mostly made of water that fills up the inside of the cell. However, while in the prokaryotic cells, we didn't see a whole lot of stuff hanging out in the cytoplasm, there are many, many more structures suspended in the cytoplasm of eukaryotic cells compared to prokaryotic cells. And we'll look at each of these and their functions. Probably the most important structure found in eukaryotic cells is the nucleus. The nucleus contains the eukaryotic cell's DNA and protects it. So unlike prokaryotic cells where the DNA just sort of free floats in the cytoplasm, the eukaryotic nucleus is bound by a double-layered membrane called the nuclear envelope. And inside of the nucleus, there is a smaller site called the nucleolus, which is where the cell's ribosomes are created before they are sent out of the nucleus into the cytoplasm. And the microscope image at the right shows you a cell with a nucleus, but to kind of help you find your way around this image, in green, I've just highlighted the cell as a whole. In blue, we have the nucleus inside of the cell. And then the darker spot that I have circled in yellow is the nucleolus. Now, in eukaryotic cells, the nucleus is linked to something called the endomembrane system, which is a collection of structures bound by phospholipid bilayer that all work together to produce materials that the cell needs, and then transport those materials around the cell. So working outward from the nucleus, the first endomembrane structure that we encounter is called the endoplasmic reticulum, or the ER, as it's abbreviated for convenience because endoplasmic reticulum is quite a mouthful. The ER is a series of interconnected tubes that modify proteins and produce lipids. And the ER comes in two forms, and the form of the ER dictates its function. There is the rough endoplasmic reticulum, which is called rough because there are ribosomes embedded in its membrane. And then under the microscope, those embedded ribosomes give it sort of a speckled appearance that looks like it would have a rough texture. And then there's the smooth ER, which does not have embedded ribosomes. So its texture is smooth. And it should make sense to us that given what we know about the fact that ribosomes produce proteins, the rough ER's function is to process proteins produced by its embedded ribosomes, while the smooth ER has a function unrelated to the processing of proteins. The smooth ER contains enzymes that produce lipids for the cell. And just a quick note, while we're on the topic of ribosomes again, all eukaryotic cells have ribosomes, just like all prokaryotic cells have ribosomes. Those ribosomes in eukaryotic cells can be embedded in the membrane of the rough ER, or they can also be free-floating in the cytoplasm, which is the form in which they're found in prokaryotic cells. So eukaryotic ribosomes can be either free or attached. And also another thing to note is that while both prokaryotic and eukaryotic cells have ribosomes, the ribosomes in eukaryotic cells have a different physical structure than the ones in prokaryotic cells, so they're not totally identical to each other. Okay, getting back to the endomembrane system, the ER processes proteins and produces lipids for the cell depending upon its form, but then what happens? So those proteins and lipids will get discharged from the ER in vesicles. And remember that vesicles are those little pockets of membrane material that bud off of the cell membrane. They can bud off of the membrane of the ER too. And it will send those vesicles to the Golgi apparatus. The Golgi is another part of the endomembrane system that consists of a series of flattened sacs. And within those sacs, the lipids and proteins that were transported from the ER are sorted, tagged, and repackaged to travel elsewhere in the cell. So in this illustration, the Golgi is shown in this sort of burnt orange color, and here is what the Golgi actually looks like as seen under the microscope. Another component of the endomembrane system is lysosomes. Lysosomes are a special type of vesicle that contains digestive enzymes that can break things down, because sometimes the cell needs to break down materials into smaller pieces inside of the cell. And this can include waste molecules, it can include toxins, even invaders. So viruses or other pathogens that invade the cell can be broken down in lysosomes. So lysosomes will take those things, disassemble them, and then they will undergo exocytosis to dump their remaining parts outside of the cell. And previously we saw little cartoon animation of what that exocytosis process looks like. But here is what it actually looks like under the microscope. So this is like a series of snapshots of it happening. We can see the lysosome on the left standing alone, and then gradually it fuses with the cell membrane, and then the materials inside of the lysosome are dumped outside. Now that covers all of the structures that are part of this so-called endomembrane system. So to recap, I have a couple of checkpoints for you pertaining to human diseases that occur when the endomembrane system is malfunctioning. One of these is non-alcoholic fatty liver disease, which is a condition where fat builds up in the liver, not as a result of excessive alcohol consumption, which is another common cause of fatty liver disease, but as a combination of diet and genetic risk factors. So in non-alcoholic fatty liver disease, there is a dysfunction in the enzymes that produce lipids in the cells of the liver. So you tell me which of the organelles within the endomembrane system is malfunctioning to result in this disease. And then next, Tay-Sachs disease is a disease where nerve cells are gradually destroyed over the course of the first few years of a child's life, eventually leading to death. It's a terminal illness. The reason why the nerve cells die is because children affected by this have a genetic mutation that prevents their brain cells from breaking down and expelling a neurotoxin called GM2 ganglioside. And as that neurotoxin accumulates, the nerve cells die and the brain shuts down. So which of the organelles in the endomembrane system is malfunctioning in Tay-Sachs disease? Okay, so up until this point, our exploration of eukaryotic cells has focused on this grouping of organelles that are collectively called the endomembrane system. But there's more to eukaryotic cells than just the endomembrane system. Eukaryotic cells also have other internal structures, one of which is vacuoles. Vacuoles are membrane-bound sacs filled with water and other stored materials. Not all eukaryotic cells have vacuoles, and in different types of cells, the vacuoles can look quite different. The cells of plants, for example, have one very large vacuole called the central vacuole, which is shown in this microscope image as the big, large, gaping, empty-looking space in the middle of the cell. It's not actually empty, though. It's full of water. And the central vacuole allows plant cells to regulate their water content. So depending upon how much water the central vacuole has taken in, it can sometimes be so large that it composes up to 90% of the internal volume of the plant cell. Other types of cells lack a central vacuole but have another type of vacuole called a contractile vacuole. So this type of vacuole is most commonly found in protists, which are the single-celled microscopic eukaryotes. And it regulates the water content by pumping water in and out of the cell. So here we see a video of our friend the paramecium once again under the microscope, and we can see his contractile vacuole at work. It's contracting and expelling water outside of the cell. Just a couple more organelles to talk about in eukaryotic cells. Our next one is chloroplasts. Chloroplasts are found in all plants as well as some protists, not in fungi or animals. The function they perform is a process called photosynthesis. You may have heard the term photosynthesis before. It is a series of chemical reactions that capture the energy of sunlight and turn it into sugar molecules. And then the plant can feed on those sugar molecules for energy. We will talk about this process of photosynthesis more in our next lecture. In this microscope image, you can see a collection of plant cells. And then within the plant cells, there are little green dots. Each one of those little green dots is a chloroplast. Another interesting fact about chloroplasts is that they contain their own set of DNA that is separate and distinct from the DNA found in the cell's nucleus. Why is that the case? That's kind of weird. We're going to talk about that in just a second, but before we do, there's one more organelle to introduce, and that is the mitochondria. So like chloroplasts, mitochondria are also important for the energy generating capabilities of eukaryotic cells. They are the site of a process called cellular respiration, which we will also be learning about in our next lecture. Cellular respiration is a series of chemical reactions that break down sugar molecules and extract their energy. Mitochondria are found in virtually all eukaryotic cells, plants, animals, fungi, because without them, the cells wouldn't be able to obtain energy. And like chloroplasts, mitochondria also have their own set of DNA that is separate from the DNA in the nucleus. So why do these particular organelles, the chloroplast and the mitochondria, have their own set of DNA? This is explained by a theory that biologists have developed called endosymbiosis theory. What endosymbiosis theory explains is that chloroplasts and mitochondria originated long, long ago in the evolutionary history of life when one cell engulfed another smaller cell, and then the two went on to develop a symbiotic, mutually beneficial, mutually dependent relationship. In other words, mitochondria and chloroplasts used to be free-living prokaryotic cells that got swallowed up by a eukaryotic cell, and then both cells decided that the arrangement should be permanent. The mitochondria and the chloroplasts were providing energy for the host cell, and the host cell was providing protection. And over time, genetic changes took place in the DNA of both cells so that now they are fully dependent on each other and they could not be teased apart. They can't live independently anymore. Now, we say that endosymbiosis is a theory, and you may remember from chapter 1 that in science, the word theory is an explanation that is considered to be as close to the truth as you can possibly get, because there's such overwhelming evidence to support it. So in the case of endosymbiosis, this evidence includes the fact that both mitochondria and chloroplasts contain their own independent set of DNA. And if you compare that DNA to the DNA of some current free-living prokaryotic cells, there are striking similarities that indicate that current free-living prokaryotic cells are essentially the distant cousins of mitochondria and chloroplasts. So the species that you see in the image on the screen is actually the most closely related free-living bacterium to the chloroplast. This species is called Gloeomargarita lithophora. But the evidence supporting endosymbiosis theory does not strictly rely on the DNA. Mitochondria and chloroplasts also contain their own set of ribosomes. And those ribosomes have the same structure as prokaryotic ribosomes, not eukaryotic ribosomes. Both of these organelles are also about the same size as a prokaryotic cell, and they are susceptible to antibiotics, which are the medications that we take to kill bacteria, which of course are prokaryotic cells. So, taken together, all of this evidence is a very strong indication that mitochondria and chloroplasts arose as a result of endosymbiosis. Now, in this checkpoint, I would like to challenge you to identify the type of transport that would have mediated this endosymbiosis event. So in other words, when prokaryotic cells were originally engulfed by the larger cell and brought inside, what type of transport was that? And be as specific as possible in providing the terminology here. We are so very close to the end now of our discussion of eukaryotic cells and the chapter in general. There are just a few more structures we need to talk about, and these structures are not organelles. They're not bound by a membrane, but they are nonetheless very important to the function of eukaryotic cells. And the first one is the cytoskeleton. The cytoskeleton is exactly what it sounds like. It's the skeleton of the eukaryotic cell. It's a network of protein fibers that runs throughout the cell, and these fibers support the shape of the cell. They keep organelles in fixed locations, and they facilitate the movement of materials around the inside of the cell. So in this microscopy image, we see a number of cells where the cytoskeleton has been artificially stained with a green fluorescent dye, and the nucleus of each cell has been dyed blue. And last but not least, we have the cell wall. So we saw that in prokaryotic cells, there's a cell wall. Eukaryotic cells can have a cell wall too. In fact, the first cells ever identified by Robert Hooke under the microscope were eukaryotic cells, specifically plant cells from the cork tree, and what he was looking at was the cell walls which you can see in this separate image. So like in prokaryotic cells, the cell wall in eukaryotic cells provides the cell with protection and maintains its shape, but the cell walls in eukaryotic cells are made of a different type of material than prokaryotic cells. So in bacteria, we saw that the cell wall was made of something called peptidoglycan. In plants, it's made of cellulose, and in fungi, the cell wall is made of chitin. We mentioned both of these substances when we were talking about macromolecules back in chapter 2. The cells of animals and most protists do not have cell walls at all. So this, of course, includes human cells. So our cells don't have cell walls. And this actually leaves our cells in danger of things like water toxicity, where they can swell up and burst and die. Cells that have cell walls can avoid death by water toxicity because the cell wall prevents the membrane from expanding outward to the point where it might burst. To finish off this lecture, I have just a couple of checkpoints for you. This first one will allow us to review a little bit of what we learned back in Chapter 2, Part 2. Cellulose and chitin are plant and fungi cell wall materials. What type of macromolecule is each of these materials? What type of macromolecule is cellulose and chitin? And then lastly, we spent lots of time elaborating the distinct features of prokaryotic and eukaryotic cells, but in this checkpoint, I'd like you to think back and identify some similarities between them. What are 3 things that prokaryotic and eukaryotic cells can have in common with each other? And with that, we've come to the end of chapter 3.